Good afternoon, and first of all, let me thank the organizing committee for having invited me to this fantastic meeting. I saw the agenda is really each topic addressed by talented speakers deserves a long time. So this agenda is very dense. I'm sorry not to be there in Munich, which, by the way, is a very lovely city, lively and full of modern art. Of course, you don't need me to go through the aging mechanisms, all the aging factors depicted in the upper left quadrant, but just to simplify the issue, I would draw the attention to the main lines of the effectors P16, RB, P53, P21, and other factors, all conjuring up promoting senescence. But senescence may be ideally a healthy senescence with age here below on the right side, healthy senescent cells, but we know unfortunately that most of the mankind goes through deleterious senescence, which means SCAPs, senescent cell anti-apoptotic pathways, senescence associated secretory pathways, and other aging factors, all basically speeding up the aging process. And accelerated aging, we do know for certain, is a background milieu of all chronic diseases affecting in modern times all of us. And hopefully not all of us, but many of us. Senolytics have certainly understandably conquered the first row on the stage under the spotlight, and this study coming from one of the big groups working on senolytics. I believe this paper comes from Kirkland, from Mayo Clinic, says that by using intermittently senolytics, DQ stands for the statin quercetin, orally administered to either senescent cell-transplanted mice or naturally aged mice. They do yield significantly increased survival by 36% and reducing mortality hazard to 65%. Many of you probably are aware this great article appeared on Science in 2019, where SASP was indicated as the culprit of accelerated aging. And inside this article, there is this beautiful picture, and the mouse on the right side is not an aged mouse under Photoshop, but is a real mouse, rejuvenated mouse somehow. But more interestingly, except besides its external outlook, is that internally key organs like kidney, heart, muscle, cataract, the brain, show sign of young-like vitality. This is really remarkable. This is an open access paper. I suggest you to look at, because you can find all the clinical trials either completed, currently ongoing, or planned at the time of publication, of course, dealing with senolytics application amenable to clinical application. This article is also important because it draws a clear line between xenomorphics, xenomorphics of things, xenomorphics, those compounds acting as SASP inhibitors, as opposed to first generation senolytics, which do target the underlying cause of detrimental SASP factors, meaning they do eliminate senescent cells. And this can be done on an intermittent schedule, following the strategy of a hit and run. So what we have been doing for the past years is to gather a lot of Atlantic fish and working on clusters of lipopeptides. Eventually, we found one cluster which we thought could be, from a preliminary work, could be amenable for a senolytic action. We did complex this cluster with a specific bioactive fraction from rhodiola, and we tested it on stem cells in vitro. You can see the dose response um, pro-proliferation activity up to 0.1 ml, but at the higher concentration, 0.1, 0.5, we had a contrary action. So we are working on the understanding of this phenomenon. On the right panel, you see how the ECM look like in our batch, and you see that by adding senolytic to our stem cell batch aged by aging mimicking oxidative stress model. The ECM were quite disassembled, yeah, 24 hours, and keeping on co-culturing them with our phytomarine senolytic, you can see the very nice realignment. It is quite remarkable senolytic effect, I'd say. Of course, pushing proliferation may not be a good idea for cancer. So when writing a chapter for this book, you see on the left panel, we dropped a few lines on a separate study where we co-cultured several lung cancer lines with our senolytic, and we found out that there was no effect whatsoever in terms of cancer promotion. And when we look at the enzymatic activity of our stem cell um, uh, injured by oxidative stress in vitro, the LPO activity was, of course, quite high, as you see in red, but the co-culture with the phytomarine senolytic significantly mitigated the oxidative stress, enhancing SOD activity and normalizing basically the hyaluronidase activity, which you know is quite active during COPD models of chronic obstructive pulmonary disease models. On the vital genes side, we had a very striking one upregulation, a fair but significant upregulation of clotho-1, SERPINA6, okay, downregulation of MMP9, a downregulation of MMP2, but upregulation at a high dosage of our phytomarine. So we also are now investigating why this happened and how to interpret the data. Citrulline, of course, has a crucial importance in aging. You don't need me to remind that. To mention one, citrulline 1 by deacetylating lysine 310 brings about a proper proteasomal degradation, whereas in those models where citrulline 1 is the deficient, the P65 is freed entering the nucleus and upregulating Syndecam 4, which is certainly not a good news because Syndecam 4, as you see, the central crossroad role in the inflammatory oxidative stress aging mechanism. The in clotho we had in our in vitro study was further corroborated and improved, I mean, methodologically by this group, which did use in mice and in humans the sampling quercetin orally and measuring a clotho urine, which went up, okay, in kidney, and also in brain. So chlorpo one is certainly a good card to play when dealing with senolytics. In our in vitro study, mesenchymal stem cells under senolytic gave rise to very good chondrogenic differentiation, and this may be of some interest considering the work that now is ongoing on improving the tropical, the dysplastic intervertebral okay, in vivo study. And this is the in vivo study we did on biopsy. We induced a COPD mimicking LPS model. The LPS was given at a dosage not to be lethal, was given for 28 weeks. Then we stopped for 1 week, and after 1 week, we split the two mice in two groups. One group was given starting to see the physiological recovery, if any, and the other group was given, besides the food, of course, like the first group, was given also the orally the interestingly, we found, of course, a very remarkable anti-inflammatory activity with reduction of elastase, reduction of malonyl peroxidase, increase of SOD, but more remarkably, from the functional viewpoint, if we see the oxygen saturation in healthy mice depicted in red, so the healthy mice are in red, the blue mice were given LPS, so where the injured mice, you can see that those mice under LPS after 1 week dramatically dropped the oxygen saturation, went even further down. Then after we stopped after 28 weeks, the supplementation of LPS, okay, you can see that those mice not eating anything except just observation and food did not recover the oxygen saturation, meaning that subacute model went really chronic, whereas those mice supplemented with peptide in rhodiola, phytomarine, let's say, senolytic, went gradually improving oxygen saturation up to basically normalizing. This is remarkable data. This really reestablishing a chronically damaged function. Very remarkable data. We were very enthusiastic about that. At the same time, our friends from our regional group were working with Kirkland in USA on gut using the sampling in the first setting, showing that it does alleviate gut inflammation through the modulation of the gut microbiota. And the same year, by chance, we were writing a chapter on this book, discussing on gut stem cells interplay with immune system and microbiota. And this is not by chance, because as you know, all this calorie restriction, the fasting regime enhance the gut stem cells. And who knows that restriction regimes, together with proper viable senolytics, not chemical, ideally, may be very nice synergy for vitality. Let me tell you now that my time is over. So I thank you for your tolerance, and I'm very also grateful to the late Professor Montagnier, very good friend, the HIV Nobel pioneer, who was always full of great ideas. So thanks a lot, and I hope the meeting will finish as it started. Great, absolutely great. Thank you. Bye bye.